Okay, so today I'm going to be talking about the development of our new risk of bias tool for network meta-analysis. We call it the RoB NMA tool. So I just want to say that we have received a grant from the CIHR, and I have no conflicts of interest to declare. And I'd also like to acknowledge the traditional ancestral and unceded territory of the Musqueam, Squamish, and Tsleil-Waututh nations on which I operate, and I'm in Squamish, British Columbia. Although I do obviously work remote. So our project team consists of a steering committee, and we meet about once a month, and as well as stakeholders and our working group. So our steering committee is made up of Ian White, Julian Higgins, Sophia Diaz, Brian Hutton, R.G. Veroniki, Jim Wright, Penny Whiting, and Andrea Trico. We also have like a slew of students, and we also have like all these stakeholders who are going to help us disseminate. So why is it important to assess NMAs for biases? So basically, without a risk of bias tool for network meta-analysis, knowledge users will not be able to tell whether a single NMA is at low risk of bias or high risk of bias for that matter. So multiple NMAs are published on the same topic, and choosing from conflicting NMAs is impossible without a risk of bias tool. So as we see, one of John's studies here on the right, the authors found 28 NMAs on treatment for rheumatoid arthritis, all with different conflicting results. So if an NMA is at risk of bias and inappropriate methods are used, the validity of the findings can be compromised, which can then further down the line impact on patient treatment and care. So the very broad purpose of our tool is to address the degree to which review methods, or actually it's actually NMA methods, lead to a risk of bias in the results and conclusions of the NMA. We also aim to develop guidance to accompany the tool and all kinds of training materials to go along with that. So for our methods, we conducted first a methodological review to identify items related to bias in network meta-analyses for potential inclusion in the tool. And for that, we found about 22 items. We then conducted a survey to ask decision makers, users, students about what type of tool they preferred. And then we conducted a Delphi survey with experts in NMA and also a bias to select items to include in the tool. And then finally, what we're doing right now is we're doing pilot testing to draft the tool for hopefully usability, efficiency, and comprehension. So the methodological review is actually, it was submitted to systematic reviews a year ago, and only now are we actually, like they said they're going to be, publishing it, but I haven't had a further email about that, so it's been taking a while. So we're hoping that's going to be published soon. The decision maker and Delphi surveys are published in one kind of combined paper in BMJ Evidence-Based Medicine Journal. So here they are here. Um, So that's the top ones under consider. It's actually not under consideration anymore. It's accepted, and then this is the one BMJ evidence-based medicine. So what do authors mean when they say they're assessing the quality of a study? So when you read in a study that the authors assess the quality of the included studies, do they mean like they assess the quality of reporting or how well the study was conducted, for example, or whether the study was at risk of bias? A lot of the times, all of these are confounded. So reporting quality is about how well the methods and results in a published study are described. So I prefer to say, reporting comprehensiveness. And I hope that people are going to be turning to that as the kind of not the preferred nomenclature from now on. So methodological quality is about how well the research is conducted. And then risk of bias is about whether the limitations in a study can potentially bias the methods and the findings. So our tool seeks to measure bias, and as I said, other tools frequently assess issues related to all of these things. So imprecision, quality, reporting. Um, so we made sure that we just wanted to really kind of focus on that bias aspect. So here, what do we mean by bias? So bias refers to systematic error, meaning that multiple replications of the same study would reach the wrong answer on average. So bias should not be confused with imprecision, because imprecision refers to random error, meaning that multiple replications of the same study will produce different effect estimates because of sampling variation. So imprecision refers to random error, so that each time a trial it takes a sample and measures outcomes, they'll get a natural variation from the true values in the whole population. So the smaller the sample, the more variability and imprecision and so then imprecision is generally reflected in the confidence intervals around the treatment effect. So, for example, the results of smaller studies with few events are less precise and produce wider confidence intervals. So bias is also not the same as quality in study conduct. So even a well-conducted study can be at risk of bias. So for example, in studies where blinding is impossible in RCTs, so surgical trials where the surgeon can't be blinded. Or similarly, not all methodological flaws are relevant to bias. So for example, failure to perform a sample size calculation or to obtain ethical approval, from that ethics board are important markers of study quality, but they're unlikely to bias study results. And then finally, bias is also not the same as the quality of reporting or comprehensiveness of reporting. So studies rarely report the methods used in great detail. So they could have used like rigorous methods, even though those methods aren't described or written up in the published paper. So then what type of tool have we developed? So we kind of made a few conceptual decisions at the beginning. And so our tool is intended to supplement an assessment of the risk of bias in an underlying evidence review. Ideally, that would be a systematic review. And then systematic review with NMA contains many of the same steps as conducting a systematic review with pairwise meta-analysis or traditional meta-analysis. So we kind of thought that assessors should start their risk of bias assessment using tools such as RoBIS, which was developed by Penny Whiting and team, or another suitable tool, although we do recommend RoBIS, would be the AmpStar tool, who's developed by Bev Shea and team. And these are both, highly, they're very robust tools that were validated and had, good results. So we also wanted to use a domain-based structure, and we use signaling statements. So our tool can be used with in NMAs with all study designs. So not just RCTs, but also non-randomized trials as well. So we also aim to develop a tool to assess both bias in individual NMA results and also those authors' conclusions about the results. So network meta-analysis of effect estimates from primary studies can result in overestimation or underestimation of the effects of a specific intervention. For specific intervention comparisons. And comparisons really is the key word here. So for example, Chumani et al. Conducted a network meta-epi study, and they found that in the majority of the 32 networks that they analyzed, small studies tended to exaggerate the true effect estimates of the intervention, possibly due to small study effects and publication bias. So our tool will focus first on identifying bias in the results of a single NMA. So bias can also be introduced, though, when interpreting the NMA results to draw conclusions. So conclusions can include something like maybe everybody's heard of spin, which is, for example, biased misrepresentation of the evidence, perhaps to facilitate publication, or erroneous, misinterpretation of the evidence, just, whatever, because they made a mistake. I want to give people the benefit of the doubt sometimes. And then ideally, potential biases identified in the results of the NMA might be addressed appropriately when drawing conclusions. So this is like an ideal scenario. Um, Similarly, a well-conducted systematic review draws conclusions that are appropriate to the included evidence. So we want to see a balanced, conclusion, saying all results, not just the ones that are, for example, statistically significant. And then, therefore, that systematic review, including that NMA, can be free of bias, even when the primary studies included in the review have high risk of bias. So the NMA tool, so our tool had we also had one other kind of concept that we wanted to put to the tool, in that for our tool, we are only focusing in on one network within maybe what could be in a systematic review, a slew of networks that are being assessed. Um, so it's not our tool is not applied at the study level or at the outcome level, but to one single specific network within a with a very specific numerical result. So different networks will often, but not always, correspond to different outcomes addressed in the systematic review, and multiple NMAs can be reported for the same outcome. So as examples, multiple networks can be reported for the same outcome due to different scales being used, different time points being used, and then also sensitivity analyses um, to look at the robustness of different effect estimates that come out of that research. So assessing a specific network result is taken because biases in a study could act unequally, and both across the study level and also the outcome level. So working on that network makes sure that one single network makes sure that we're focusing on that specific thing and assessing the bias in relation to it. So I just kind of wanted to give you an orientation to the tools out there, because we often get I bet all of you are very familiar with the tools out there, but a lot of people aren't. And so I wanted to kind of orient you to where our tool fits in with to this framework. And so the figure shows on the left, basically, tools that are used by authors of systematic reviews containing network meta-analysis. And then on the right, it's people that want to critique, the NMAs that have been published. So authors of NMAs use tools to assess bias in the included studies, such as you could see here, the Risk of Bias 2 tool by Cochrane, or the RoB NMA tool, which is for missing evidence, or, for example, the Grade NMA or the Cinema tools, which are used to assess, the pooled primary study evidence. And then once the study is finished, they'll use, Prisma NMA to assess reporting comprehensiveness. And then finally, when it's all published, reviewers and NMA assessors, critiquers, people who want to see what the biases in the network meta-analysis would first use the robust tool, which we recommend, and then eventually we'll be using, hopefully, our RoB NMA tool. So we have written an elaboration and explanation document, and we developed guidance which explains the background, the mechanism of bias, and for each item, and how to assess each item based on each response. So our responses are true, probably false, and no information. We do have a website under development, and it will contain lots of training materials. So this is just a preliminary look at the structure of the tool. So there's 3 domains for the tool, namely interventions and network geometry, effect modifiers, and statistical synthesis. In total, there's 17 items across 3 domains. And after answering the signaling statements in each domain, the assessor then answers 2 overall judgment statements. And then at the end, finally makes an overall judgment based on low, high, or actually low, high, or some concerns. So our tool works by using a set of reasonably factual signaling statements to help authors determine bias within these three domains. Um, and so we have true, probably true, are considered to be pretty much the same, just expressing different levels of certainty in the response. No information is used. It should be used quite sparingly and only when it's pretty much impossible to make a judgment of yes or no. Um, we also encourage authors to record, the reasons for their judgments. And then for the domain level judgment, the signaling statements then lead assessors towards the domain level risk of bias judgment will be, and which was judged as low risk of bias, some concerns, or high risk of bias. So that's the judgment for each domain here on the right. And then for some concerns, the some concerns is basically replaces unclear risk, which is found in other tools such as RoBIS. And then here at the end there, we have the final overall judgment. And that again comes down to two judgments that you can make, which is basically no concerns. Sorry for the risk of the results section, we have high risk of bias, low risk of bias, and some concerns. But then for the conclusions, we just made it very simple. You either have no concerns or concerns for the final conclusion overall judgment. And then I'm just going to give you just examples of three items in the tool for going down to each domain because I don't have time to go over everything. So for this domain one, interventions and network geometry, we have 4 items in total. The first ones is all interventions and their comparators included in the NMA are reasonable alternatives for the whole target population. This is about joint randomizability. The second one is no interventions were inappropriately excluded from the network. Interventions were appropriately grouped into the nodes in the network. And then all compared interventions were connected through a suitable chain of study comparisons. So here note that we're not concerned about the interventions included in the network and not the studies. So this is not a study level assessment, but an assessment at the level of the comparisons, okay, of the interventions. So these 4-item assessment should be based on, I want to mention again, one single NMA result within the review. So this is my example item. So I picked item 1.3, which is interventions were appropriately grouped into nodes in the network. On the right, you see what are like a portion of what our E&E is, our elaboration and explanation guidance document. So the grouping of interventions or specific implementation of interventions into nodes is often required in a network meta-analysis. So this applies similarly to grouping of any control interventions, including the network, including placebos, no treatment control, or usual care. So for example, if studies from 1980 to 2023 are included, they might have very different definitions of standard of care, and you might, the NMA authors might want to have considered that, and we want to see that reflected perhaps in the description in the manuscript. So when interventions are grouped, it's important that they sufficiently are similar so that they're not expected to differ in their relative intervention effects. So bias may arise in this case, arise for the target question if the effect estimated by the combined interventions does not reflect the effect that would be observed if the target intervention um, uh, not combined had been used. So for domain 2, here we have 4 items. Outcome definitions and time points were similar across the direct comparisons in the network. Effect modifying participant characteristics were similar across direct comparisons in the network. Effect modifying study characteristics were similar across direct comparisons in the network. And then, so finally, this kind of like overall summary question is if you've answered, if you have concerns in from 2.1 to 2.3, so if you do suspect effect modifiers, then you would answer the analysis. Did the analysis appropriately address the differences in effect modifiers across the network? Oh, sorry. So here I chose item 2.3. So effect modifying study characteristics were similar across direct comparisons in the network. So effect modifying study correct characteristics can vary across studies for and across comparisons for a particular comparison. So it's the same with standard pairwise meta-analysis, and but also they could also vary between comparisons, so which causes inconsistency in the network. So, for example, features such as study design, for example, a cluster randomization or run-in period, study setting, such as primary versus secondary care. Like one good example that I thought of yesterday is, study setting would be, in vaccines, for example, there might be a seasonal effect, so summer versus winter. So that would be kind of go under here as well. So if there's an imbalance in the distribution of effect modifiers between different types of direct comparisons, the NMA will be biased. So for example, if studies comparing intervention A with intervention B are mostly conducted in primary care, and then studies of B and C are mostly conducted in secondary care, then there'll be an indirect comparison of these results to estimate the intervention effect of A versus C, and this will likely be biased for either of these settings. If the intervention effects systematically differ between settings. So this is due to basically the failure of transitivity or consistency to hold across the different sources of the different comparisons across the network. So here's domain 3. So it has 9. And so yeah, first one is all eligible results were included in the analysis. The third is all predefined analyses, and only those analyses were reported or departures were explained. Biases in primary studies were minimal or addressed in the synthesis. Appropriate methods were used to handle multi-arm studies. Appropriate assumptions were made about homogeneity or heterogeneity of effects within comparisons. There was no evidence of conflict between direct and indirect estimates of the same effect. And then that's the next question goes on to that. So if you've answered false, then conflicting results between effects and indirect evidence was adequately addressed. 3.8, if a Bayesian analysis was performed, the choice of priors was appropriate. And then 3.9, sensitivity analyses demonstrated that findings were robust to the statistical model and all estimation methods. So the one that I chose was item 3.7. So conflicting results between direct and indirect evidence were adequately addressed by the authors. So NMA is not valid when the direct and indirect evidence are not in agreement. Conflict between direct and indirect evidence implies that the fundamental assumption of transitivity in an NMA does not hold. Sorry, I'm just going to drink something. My throat's a bit sore. So the conflict between direct and indirect evidence may lead to bias in the network results. So when the direct and indirect evidence for a comparison do not agree, there's inconsistent information about that comparison. And depending on which source of evidence is nearer to the truth, in parentheses for this comparison for the underlying research question addressed by the network, and on the relative amount of information in each source, the results of this comparison and other comparisons informed by it can be biased. So here, note that in the synthesis domain, we don't have an item related to intervention rankings. And this is because if you do identify bias in any of these 17 items, that bias is likely reflected in like these summaries, these sucra summaries or these ranking summaries. One of the examples is sucra when you're doing a Bayesian analysis. So Robust is a new tool. It's relatively new, I guess. It's getting older every day. Designed for those appraising reviews, systematic reviews especially. Um, the Robust tool asks assessors to make a domain level judgment of risk of bias. I'm just gonna have one slide on that just to show you what it's like. There's four domains in this tool. It's study eligibility criteria, identification and selection of studies, data collection and study appraisal, and synthesis and findings. So if you're using the Robust tool in conjunction with our RoB NMA tool, you would kind of drop this synthesis section actually. So each domain contains signaling questions with answers being yes, probably no, no, or no information. And then after answering the questions, you make an overall judgment, which indicates your concerns about the methods relating to each domain. And for that, the responses can be low, high, or unclear concerns about the methods. And then in the third phase, the assessors make an overall judgment, seen down at the bottom, by assessing the interpretation of the findings, specifically whether the concerns you identified, all the biases you identified were addressed, may be mentioned in the limitation section of the discussion, and then whether the relevance of the studies was considered, was it relevant to the review question, and then whether the reviewers avoided emphasizing results based on statistical significance. And so I just wanted to mention, yeah, that we have all those synthesis questions, so that definitely needs to be skipped in the Robust tool. And then our RoB NMA tool has an overall judgment that can be used that incorporates this Robust judgment as well. So here's what it looks like in the final phase. So you would kind of incorporate whatever your judgment is about the systematic review bias. So for example, if it was low risk of bias, you would incorporate that. And then you would use the results of all three domains and you would take consideration of any biases that happened in the results and then whether those results were adequately reflected in the conclusions if there was no evidence of any kind of spin or any other kind of misinterpretation of the results. So here I'm just going to repeat again. So assessors could be interested in whether the results of a systematic review were of one specific NMA are at risk of bias. For example, they might be just interested in one network related to one outcome and one, estimate. But they also might be interested in the overall conclusions made by the authors. So which of these, somebody is interested in will depend on the purpose of the bias assessment and whether they're using, the results are both. Yeah, that's about it. And then we are piloting. I'm encouraging all of you to reach out to us. We are going to be kind of ending piloting in about 3 weeks' time because we kind of want to wrap it up, and we're kind of planning on submitting for publishing in January, so I kind of want to get that finished pretty quickly. And we'll be doing more rounds of piloting after that, but we want to get the tool out to people. So for piloting, pilot assessors can use our tool either on an NMA of their choosing, because we want to make sure that they have some, clinical, like at least some clinical expertise in the NMA that they're going to be looking at, or they could use one that I sent to them. And then they'll complete a short user survey just to determine, what their thoughts are about the tool, if they have any feedback for us, what the mean time it was for them to do it, and especially, what types of items people find difficult to answer, because we want to try and make it as easy as possible. But one note about the tool is that it's not comprehensive. So, we can't possibly list every single method that could be used to address, the biases. And so we do recommend that people partner with, whatever your expertise in. We recommend that a statistician, a clinician, and then, maybe a methodologist kind of partner together, because then you have everybody in one room helping to make decisions about the bias in an NMA. So in conclusion, um, our NMA tool assesses the potential bias, risks of bias by identifying limitations in the way an NMA was conducted, including aspects of how the evidence was assembled that may lead to bias in one NMA's results and conclusions. Our tool contains 17 items organized into 3 domains: interventions in network geometry, effect modifiers, and statistical synthesis. And we developed guidance which explains the background, the mechanism of bias, and how to assess each item based on each response. And then we started piloting, and we welcome participants interested in testing out the tool for us. So thanks very much, and welcome any questions.